Everybody, well, welcome. I'm so excited to see so many excited faces here on our Zoom. My name is Sarah Welsh, and I am the Assistant Director at the Community Boating Center. A couple of things before we start this Zoom. So as you know, the Zoom is about the bioluminescence of the Salish Sea. Standard Zoom protocol. If you guys don't mind leaving yourself muted and maybe even turning off your cameras, that can just help with the bandwidth to make sure that everyone has good internet throughout this presentation. What you can do throughout the Zoom is to add questions to the chat. I will be helping to monitor those for Lucy and make sure that we can get everybody, get all those questions answered as quickly as possible. Additionally, yes, we are recording this Zoom. So if you weren't here at the very beginning, there is a Zoom recording going on. Another good reason to turn off your camera if you would like, but we will have this recording available later. Please feel free to direct message me or just put any questions in the chat as we go, but we're going to go ahead and get started. So first of all, this presentation is being offered by the Community Boating Center and the Marine and Coastal Sciences Lab at Western Washington University. For those of you that might have not ever been to the Community Boating Center before, we are located up in Bellingham Bay, and we are a small nonprofit fostering safe small boat recreation here on the bay. The CBC was founded by a core of local small watercrafts enthusiasts in 2006, and we first opened in 2007. CBC, Community Boating Center, is not a club. There are no memberships and no dues. Programs and services are available to all, much appreciated tax-deductible donations, season pass sales, dry moorage fees, and rentals of sailboats and human-powered watercraft. Safety and skills education is the core of the organization's mission, and volunteers are integral to all parts of the CBC. I'll be adding our website into the chat, but please feel free to look through just to familiarize yourself with some of the offerings we have, one of which, of course, is our amazing bio tours, which I'm so excited for Lucy to tell more about. In our next slide, you'll see where we're located. So for those of you that may not know, we are located here in Bellingham. And I just want to take a moment to do a land acknowledgement and acknowledge that we are residing on the traditional, ancestral, and unceded territory of the Lummi people. The Lummi people are the original inhabitants of Washington's northernmost coast and southern British Columbia. They lived in villages throughout this territory and continue to have ongoing relationships with these areas. Since time immemorial, they have celebrated life on their land, waterways, and on the traditional, ancestral, and unceded land of their people to perpetuate their way of life. Please join me in taking a moment of silence as we honor their ancestors and we acknowledge the past, present, and future of Lummi people as the original inhabitants of this land. All right. And I do want to go ahead and introduce Lucy, who will be doing this presentation. So Lucy, sorry, I just closed my thing. Lucy is one of our new interns here at the Boating Center. She's currently enrolled at Western Washington University studying the new Marine and Coastal Science major. People often ask Lucy, what's your favorite marine critter? And she excitedly can tell you all about the peacock mantis shrimp. For example, did you know they can be 12 primary colors? Additionally, Lucy likes to say that she grew up in classic P and Dove fashion, kayaking around Seattle and playing lots of soccer. Beyond being infatuated by anything to do with water, Lucy enjoys rock climbing, being a DJ for Cugs, which is a local radio station, gardening, and listening to live music. Well, thank you, Lucy. Go ahead and take it away.
Awesome. Hello, everyone. It's lovely to have so many people participating. And although I would love to give a presentation about the peacock mantis shrimp, today, a close second favorite of mine, I'll be talking about bioluminescence. All right. So what is bioluminescence, right? Do we have any guesses?
Glow in the dark.
Okay, yeah, Harry Potter magic. I love this. Plankton, ooh, that's exciting. Right, so bioluminescence is essentially the process of emitting light through a chemical reaction in a living organism. So we have some examples. I feel like the most common bioluminescence that a lot of people know, which they may not even know that they are contributors to bioluminescence, but it's fireflies. And I didn't even know this because someone who's studying in marine science, I focus a lot more on like marine animals, but I thought that was really interesting. Another example is the bioluminescence that we see around here. So this photo is taken at Ruby Beach out on the Olympic Peninsula, but the same thing happens right here in Bellingham Bay. And this bioluminescence is plankton, which is provoked by mechanical stimuli like brushing against these rocks. And that's when bioluminescence is emitted here. Yeah. All right, and then we even have things such as the deep sea marine worm or jellyfish that emit bioluminescence. And we had a book donated to the Community Boating Center. It's called Below the Edge of Darkness by Dr. Edith Witter. And has a really awesome quote in it that I just kind of wanted to summarize. But essentially, if most of the animals in the ocean are bioluminescent, then a majority of creatures on our planet are communicating using language of light dialects that we don't comprehend. I just feel like that's pretty powerful and telling to how awesome bioluminescence is. Oops. All right. So a little history, a little historical background on bioluminescence and how it's been evident in years past. Firstly, in the 17th century, George Eberhard Rumphius, who was a physician, was working on his botanical herbarium. What is an herbarium, you may ask? Oops. It's essentially a systematically arranged collection of dried plants. So it's used to ID plants and look at common characteristics for easy identifying in the future. And I was really fortunate. I just took an algae class at Western and I got to make my herbarium, and here are some examples. But back to the cultural application and the cultural story. Rumphius, this physician, recorded that indigenous Indonesians, which he traveled to work on his herbarium, often carried bioluminescent fungi to light their way through dark forests. Pretty crazy. And then another example is in World War II, a lot of Japanese soldiers used these organisms called ostracods, which are really, really tiny crustaceans, couple millimeters long. They're often described as clam shrimp because they kind of look like a mix of the two. But they would collect them and they would dry them. And then if they crushed them in a moistened palm, they would glow. So it was the perfect amount of light for reading maps and letters, but too dim for nearby enemies to spot. I have a video here, which might actually help. Just to kind of get an idea of what these ostracods look like in movement. And you can see right here in this right-hand corner, this is a magnification of 40 times, so they're pretty small. But I think it's just kind of fun to watch them move. Right. Okay. Oops. Sorry about that. Okay, so we know a little bit historically about how bioluminescence is used and these historical and cultural applications, but how do people use bioluminescence in modern times? How do scientists use it? So there's actually been a lot of research done on it medicinally. And in this one species of jellyfish called Acora victoria, which is found in the Salish Sea as well, was discovered by Osamu Shimura in the 1960s of having this green fluorescent protein. This diagram's a little confusing, but essentially he won this Nobel Prize in chemistry in 2008 for this discovery. And he found that when this jellyfish was exposed to UV light, it emitted this green fluorescence. And biologists found a way to extract this green fluorescent protein and used it as a marker protein. So a little bit of background, going back to those like basic biology days, we need to know a little bit about DNA expression. DNA expression is essentially how in our human bodies, all of our cells have the same DNA, right? But they differ in expression. So what makes skin cells, and muscle cells is that they're expressing different genes that they have. Skin cells have the genes for muscle cells, but they're only expressing those for skin cells, which is what makes them part of the skin. So when scientists combine this green fluorescent protein gene to another gene that produces a protein that they want to study, if that cell produces green fluorescence, then scientists can infer that the cell expresses that targeted gene. So essentially, this has been looked at to understand cell damage in Alzheimer's and other neurological diseases, to track the spread of HIV, and even to track cancer sites. So according to this one article, green fluorescence protein was used in vitro, so in test tube, and it was found that a high level expression of green fluorescence protein provided detection and visualization of distant micrometastasis in organs that would usually not be seen in scans. So I didn't know what metastasis is, but it's essentially secondary growths at a distance from the primary site of cancer. And then there's micrometastasis, which, as mentioned previously, is undetectable by scans and undetectable by the technology we have today. So they found that if they use these green fluorescent proteins, they were able to find the micrometastasis, these small, really microscopic secondary sites of cancer. And in past studies, they've used fluorescent dyes to track tumors, but they lose fluorescence after a few generations. So these green fluorescent proteins are really exciting because they don't lose fluorescence over time, and because they're passed on each generation. All right, so we know a little bit historically, we know a little bit about how it's been used in modern times medicinally. But how are organisms bioluminescent? So in contrast to Acoria victoria, that jellyfish I just showed, a lot of organisms actually don't have green fluorescent protein and instead have this substrate and enzyme reaction. So the substrate I have labeled here is called luciferin, and the enzyme is luciferase. And when these combine, it emits bioluminescence. Enzymes catalyze reactions, so they start reactions. A basic human enzyme example, if you're still a little confused, is lactase, which breaks down lactose. So some people don't have lactase, so they're lactose intolerant. And similarly, some plankton don't have luciferin, so they don't emit bioluminescence. And just to clarify, these are generic terms used for any bioluminescent substrate or enzyme, of which there are many. So when these two combine in an oxygenated environment, it creates light. Okay, but why do organisms emit bioluminescence? It's not just for our own viewing. It's not just for some cool kayak paddling. There has to be a reason, right? So this is a little confusing of a diagram, but the thought is that bioluminescence has evolved independently more than 50 times. To break this down and so you can view it a little better, I have a close-up. And I think it's really interesting because you can see how it's been evolved so many times. The green is terrestrial or fresh organisms, and then the blue is marine organisms that emit bioluminescence. And you can see over here there's fungi, and then there's Cnidaria, which are like sea anemones or a part of Annelida, segmented worms, mollusks like snails or sea slugs, and then even vertebrate organisms with vertebrae. And so we have sharks and rays, which is really, really cool. I'll just take a second if you all want to look at that really quick. All right, So fireflies often use bioluminescence for mating. In deep sea, there's a lot of hypotheses that bioluminescence is used for communication, right? It's so dark. But what about plankton? That's the interesting one. Essentially, there are two hypotheses. The first being that when plankton emit bioluminescence is used to scare and stun predators. So think about a really dark night and all of a sudden a bright light, it's really hard for your eyes to adjust. So maybe it allows that extra second for plankton to get away. And then the second hypothesis is a little confusing, but essentially it's to attract secondary predators to eat the predators of the plankton. So if fish prey on plankton and they eat this plankton, then if they're swimming at night and this bioluminescence, this planktonic bioluminescence lights up around them, then maybe a seal can see the fish really easily and eat that. Okay, so when people think of bioluminescence, in my opinion, they often think of the sandy beaches in Mexico or the scary deep sea organisms like this anglerfish. But what does this have to do with here? You may be asking, Lucy, what does this have to do with the Salish Sea? Well, exciting enough, we have some in the Salish Sea, and that's what I hope to be studying. So kind of the two main contributors to bioluminescence around here are plankton and plankton. And within that are dinoflagellates, which is this video in the picture on this top right, excuse me, and then zooplankton. And they emit bioluminescence when provoked by mechanical stimuli. So when a kayak paddle strokes the water or when a fish swims, like I mentioned before. And plankton or dinoflagellates are single-celled organisms considered algae. And to break down that word a little, dino means whirl and flagella means whip, which just describes how they move. But one of my professors this quarter often described them swimming like a really bad football throw. I actually caught a video of one, which I will play. You can see them right here. They're these yellow things moving. All right. Oops. Okay. So we know a little bit about dinoflagellates now, but with zooplankton, they're essentially heterotrophic. So they don't produce their own food. And they're plankton that consist of small animals and immature stages of larger animals. So a little bit of a closer look on these zooplankton are, we have things such as barnacle larvae right here, and then copepods are a common zooplankton. And they're essentially small aquatic crustaceans, same group as shrimp and crabs and krill. And then right here is just another photo of dinoflagellates. Some other examples of zooplankton can be octopus larvae, coral polyps, snail larvae, all sorts of fun things. And then dinoflagellates, just to clarify, are not photosynthetic, but they do ingest phytoplankton. So they're still influenced by light levels. Okay, if you know a little bit about bioluminescent plankton, there's one that I feel like a lot of people tend to throw around. It's called Noctiluca. In Latin, this means nightlight, and it's kind of the most common bioluminescent species that a lot of people know. What are Noctiluca? They're dinoflagellates, so whirl and whip. And they are oftentimes bioluminescent, and they are in the Salish Sea. But there's one study that was done in 2019 that hypothesizes based on a few samples that the Noctiluca on the west coast of the United States actually aren't bioluminescent. So they're present here, but they lack luciferin. They lack the substrate, right? Kind of like lacking lactase and not being lactose intolerant. And so in this study, they essentially took 4 samples. They took one in Port Aransas in Texas on the Gulf of Mexico, one in Scripps near San Diego, one in Santa Cruz, and then one in Fidalgo Island, actually from Shannon Point Marine Center, which is Western Washington's Marine Center, which is awesome. But the only ones that were that emitted bioluminescence were the ones in the Gulf of Mexico. And then you can see I added some plus and minuses here. The pluses and minuses are just telling you whether or not luciferin is present, so whether or not that substrate was found. And so in the Gulf of Mexico, they found that luciferin, which is probably why it was found to emit bioluminescence. But then in San Diego and right here in Anacortes at Shannon Point, they didn't find luciferin in those Noctiluca. Some other interesting things is the Noctiluca along this west coast of the US is half the size of the bioluminescent Noctiluca found around the world and in the Gulf of Mexico. And then the highest number of mutations were actually the ones in the Salish Sea, which is really, really interesting. So my advisor, Dr. Robin Codner, she's here today. She's lovely. Both of us are we're kind of skeptical of this study a little bit. So they only looked at 4 locations, right? And it showed that in Mexico, these Noctiluca have the DNA and express it, going to that back to that expression, right? But the ones in Scripps and the ones at Shannon Point Marine Center don't express it, and we don't know why. But we're not completely convinced that not all bioluminescence on the West Coast doesn't emit bioluminescence. All Noctiluca, excuse me. So who is responsible for bioluminescence here, right? You may be asking that. Well, that is one of the questions that I hope to answer this summer. I hope to look at the community composition, who is contributing to bioluminescence, and then also how bioluminescence is changing over the summer. At the community boating center, some kayak guides have kind of come up with some non-scientific terms of how the light levels change throughout the year. So they describe it as dust, orbs, and sparkles, because throughout the summer it changes. And so maybe there's different planktonic species at play. And that's what I want to look at. So what specifically will I be doing? Well, to look at the community composition, I'll need to do a plankton tow. I have my friend Isaac here, who I think is attending tonight. And he's washing down this plankton tow. I'll let you take a look. All right, so this plankton tow is a net that you put in the water and you tow it for however many minutes or however long you want. And it's a net with a really small mesh size in order to get these plankton, right? So Isaac right here is washing the plankton down into this little capsule. It's called the cod end. And once you get it in there, then all the plankton's been filtered larger. Organisms can't make it in. And so then we have our plankton and we can look at that. And then also what I'm interested in looking at is the DNA. So if the luciferin genes are present, if they're expressing the luciferin substrate, and if it's present in different species. So Dr. Robin Codner does a lot of work with snow algae, and her student a few years ago, you can see, is filtering some water. And once you put it on these 2 towers and you pour some water in and create a vacuum, you can put it on these small filters and then do some DNA assessment. So I also really hope to be looking at light meters and luminometers to measure the light of the bioluminescence every night. We're still working on the grants and raising funds for that equipment, but it would be really exciting, because essentially if we have a night with a large amount of light, bright light, and I find a majority of one species from this plankton toe, and then we do a DNA test and we find that they have luciferin genes, then we have all this evidence that can help provide support that plankton is emitting bioluminescence in Bellingham Bay. So that's kind of how we're taking the approach. But how can you get involved? So the really, really exciting part about this project is that in collaboration with the Community Boating Center, we'll be doing citizen science. So you are able to join on a bioluminescent paddle. This QR code should give you a link to the website. And these are just some photos from paddling out on Bellingham Bay. But if you come on a bioluminescent tour, you'll get to see me do some of the research. Maybe you'll help me filter or do a plankton tow. And I think it'll be really, really exciting. And then to get you excited and amped up, I have this video from the Oregon field guide of my advisor, Dr. Robin Codner, kayaking for bioluminescence. Keep in mind, it is from 2017, so it was before the study of Noctiluca maybe not emitting bioluminescence in this area, but I'm gonna play it.
Whoa, look at that sun. Wow.
Biologist Robin Codner and her friends love a good light show. But tonight's display is not exactly what they had in mind.
This thing right here is what we need to watch. It's definitely got some funky color getting closer.
Wow. I want to get my boat because it's going to be warm inside my boat.
This is Friday Harbor in Washington's San Juan Islands. And despite the threat of lightning, Robin's got a date with some algae.
So there's this really cool group of algae that's very ancient. We think actually might be the most ancient of all the algae that's still alive. What's interesting is somebody who's an algae specialist like me only kind of calls these guys algae. So they're related to things that do photosynthesis, but they actually don't do photosynthesis.
What Robin's talking about are protists, a kingdom of life forms that aren't really plants and aren't really animals.
And what they do is they eat other algae that do photosynthesis. And the other really cool thing about them is they're super diverse. Maybe the most diverse group of organisms that we know about. So that makes them super successful.
But these single-celled organisms are tiny, and they're not always easy to find.
This is my first time paddling with lightning in the background. We are looking for the Noctiluca. The Noctiluca is a great name because Noctiluca in Latin means nightlight.
But so far, Noctiluca's not really living up to its name.
One of the reasons why we might not be seeing it right away, even though it's getting dark, is that Noctiluca actually has a circadian rhythm. So we have this like internal clock. It knows if it's night or day. That's one of the reasons why even now at dusk, we're not really seeing it yet.
Well, Noctiluca wakes up to the fact that it is nighttime. Let's take a closer look at exactly what it is.
So we're going to do a plankton tow here off the outer dock.
Plankton, in case you forgot your high school biology, is a catch-all name for a variety of teeny tiny plants, animals, and other life forms that live in the ocean. And Robin's got a special net for collecting it.
So this net has 30 micron holes in it. There's a lot of the phytoplankton that are smaller than that, and they'll go out of the net. But the Noctiluca that we're after, that's going to be way bigger than the holes in this net. And so it's going to be easy for those guys to get trapped in here.
Remember, we're talking relatively way bigger.
We can see them under the microscope really easily when they're easy to identify. The very first thing I do when I collect a water sample is I take it back to the microscope and see who's there. It doesn't look like it's very much area to cover, but microscopically, it's actually a lot of area. Oh, look at that. It's a little larvae. It's not Noctiluca. Noctiluca is not this charismatic. There are tons of exciting things in here. See this guy swimming around? That is also not Noctiluca. The hunt continues.
Among all the many residents
There's copepods in here.
of this single drop of water,
Bunch of little worms. There's a diatom swimming away.
finally,
Whoa,
Robin finds the tiny,
it's totally it, I think.
single-celled organism
So I found it.
at the center of her passion for microbes.
So these are the Noctiluca cells. You can see a little tail moving around.
A closer look reveals tiny organs lining Noctiluca's cell wall.
And they're called symptomins. Inside of those, there are the chemicals that when they come together, produce the light because they need to disturb the water to get that chemical reaction happening because that's when the light is emitted.
Which brings us back to that nighttime paddle. Remember how Noctiluca means night light?
Oh, wow.
It's also called by what it does.
The bioluminescence we see is coming from right inside the cell. All we need to do is move the cell around a little bit so that it's shaken up.
When she's paddling inside the water, there's like this wake of sparks. Oh, it's so cool, man.
But picking it up on camera
I will see video working. It seems like we can't crank the camera high enough.
requires some on-the-spot improvisation.
We're going to switch over to photographs. The exposures are slightly longer, which is what we need in order to draw the light out from the bioluminescence because it's just not quite bright enough for video.
Pretty cool. Looks bright.
Oh yeah.
But why that light is produced isn't really clear.
Why would the organism invest all that energy in making the light? And it's not just to give us a light show, it's because we think there's an evolutionary advantage.
In other words, Noctiluca's ability to produce light may help it survive.
It's when a predator swims by and moves the water around that the light will happen. And so the reason that we think it does bioluminescence is to distract its predator so that it can, swim away or the predator is going to go away.
There are also theories about why Noctilucas show up all over the world, but appear to like some places more than others.
These are all questions that are like right on the forefront of understanding microbial ecology. And I don't know that we really have a good answer for that. So every time you go out to the ocean and you're looking for something new, with microbes, you find something brand new.
Wow.
While Noctiluca's not giving up all its secrets yet, what is clear is that when you get a few million of them together, the conversation that ensues is extraordinary.
You ever seen the movie The Abyss? Boom.
Yeah.
There it is right there.
It's almost like if you take a stick out of the fire and wave it in the air and the sparks fly off of it.
It's this way that these microbial communities really act together, almost like a single organism. We think of them as being so simple, but they talk to each other and they send all these chemical signals back and forth. So they really can have major effects on how the others behave.
Just need lightning strikes to not come down on us.
Kind of nice ambient light show.
Light in the water and light in the sky.
Yeah.
All right. So just want to say thank you to a few people. Dr. Robin Codner, Dr. James Diamond, the U.S. Sailing and Reach Program. And then I think we're ready for some questions.
Hello, everybody. Lucy, there's some questions in the side column, which I can read them for you, but we'll just kind of start out at the top. One person did ask if we put, I think talking about the proteins, if we put them in meat, can I glow in the dark? And I think you did kind of answer that question about how scientists use those different, yeah. Someone also sent some websites that were helpful that just kind of backed up what you were talking about as well about putting the bioluminescence into various tumors and things like that. Anastasia had a good joke. And let's see, if they're injected into the body and don't seem to degrade like the fluorescence did, what happens to them when the testing purpose for them is over? Are they excreted or do they remain in the body?
This is a great question. I mentioned this briefly, but the these studies were only done in vitro, so they're only done in like a lab and a test tube, and they haven't been tested. As far as the study I looked at, they haven't been tested inside someone's body. So I don't entirely know how that would work.
Fair.
Yeah.
Stephen
That's
asked-
true. Yeah, and when they do it on humans, they only do it in cell culture. So it hasn't actually been put into any person or any vertebrate for that matter.
Perfect. And Stephen asked, what are the size of these organisms? I believe talking about the dinoflagellates.
Yeah, Robin answered that right below, but the Noctiluca are usually about like 1 millimeter in diameter.
Does the stage of the moon affect the amount of light the plankton emits?
I'm not sure. I'm not sure how to categorize moods in plankton, but it is evident that not a lot of studies have been done on them. So we're definitely answering a lot more basic questions this summer. But yeah, that's a funny one.
How does water temperature affect the presence of bioluminescent organisms?
That's also a good question. I don't know specifically on water temperature. Maybe Robin has a better answer. But I know that as light increases, so sunlight, we see a lot more bioluminescence, and we see them especially in the summer. And that's because they're influenced by these phytoplankton, because they feed on them too. And so if there's more light, there's more phytoplankton, and then there's more food for all the bioluminescent organisms.
And then we did have an autocorrect that it was supposed to be moon, not mood. I liked both questions though. So how does the moon phase, which that makes sense, affect the visibility of the bioluminescence?
Right. So if there's a brighter moon in general, there's a lot more light out at night. So you're not going to pick up on the light as much. So that's why one of the best times to go out is on a new moon or when there's no moon. Yeah.
Perfect. Is there bioluminescence at the bottom of the Mariana Trench?
Probably.
I think in that book you referenced near the beginning, the woman that wrote it, she went down, and I forget how deep she was, but I want to say it was like 1,000 feet, and just talks about how it looked like fireworks around.
Yeah.
Okay, so Brady has a question. Super interesting talk, Lucy. Will your net capture calanoid copepods? And if so, will you survey them for luciferase and whether they are a dominant bioluminescent group in the Salish Sea?
Dr. Brady Olson was my professor this past quarter for my algae class and for marine ecology. But embarrassingly, I can't remember what calanoid copepods are. So I'm not sure.
I'm going to help you out and say they might if they're at the surface, because we're planning on doing surface tows. So, and there was another question in the chat asking about plankton migration, and some of the plankton do migrate from the surface waters and down deeper during the day, and then can move back up, and that includes some of these dinoflagellates. But what's going to be, we have to figure out the methods for towing the nets with the kayaks at night. And so it's going to be and the bioluminescence is on the surface at night when we will be bioluminescent kayaking. So we will primarily be surveying surface waters at night. So we may get some calanoid copepods. The primers, and there was another question about how we're looking at the DNA using PCR, and we are using PCR, and we have dinoflagellate specific primers for the luciferin gene. So the primers that we are going to be working with would not allow us to assess bioluminescence in copepods, but we could do that if we wanted to expand our study.
Perfect. One of our next questions is, where are the best places to see bioluminescence in Bellingham Bay? And just a friendly shout out to the Community Boating Center. We are starting our tours this week. So if you want to join along with us and help Lucy collect her data, that's a great way to do it. From shore, I'll just answer this really fast. Teddy Bear Cove has a great place to kind of splash around. But definitely consider joining us out on a tour. There is nothing more magical, I think, than seeing it from a kayak. It's really fantastic. The next comment I see is, thank you, Lucy. You are the very coolest. Let's see. Looking forward to hear what we find. As a kayak guide, I've noticed warm air, cold water, and less would make for bright bios. Nice, Christine. And then we talked about the plankton migration and the ATPs. Where is the presence of bioluminescent organisms most prominent? Are they present in fresh water?
That's a good question. I'm not sure about fresh water. But yeah, bioluminescent organisms seem to be all over the world. Most prominent, maybe like most easily seen, is in the deep ocean, I would say. But yeah.
Very good. Jasmine mentioned that she missed the beginning of this talk and is wondering, we do have this recorded, and I learned during this chat that we had 100 people max, so that's why you never saw the number go up over 100. So there will be a recording to find online. I think more than half the people missed it, unfortunately, so I apologize to everyone who got, but we will put this online, and maybe we'll have Lucy do another one. In the lab, when studying these plankton, do all the lights have to be off?
That's a good question. So at least for the research that I have figured out right now, when we're looking at the DNA and we're looking at the plankton, it's a little difficult to look at exactly which plankton species are contributing to the bioluminescence. So that's why we're using all these different resources, like measuring the light levels and then seeing which plankton's the most present in the sample, and then also looking at the DNA and if there's luciferin. So I think in a lot of high technology places, they probably have a method to have the lights off and mechanically stimulate the plankton to see which ones are emitting bioluminescence, but we're going about it with a different approach.
How long can a dried ostracod produce light?
It was really interesting actually that you mentioned that, because in that study, it was this specific bioluminescent researcher, and they met with someone who had collected some of those ostracods. And I think they said something like it was 50 years later and they still could just put them in like a moistened palm and they would glow.
Wow, that's nuts. One of the questions is anyone marketing the two historic uses? I don't know. Don't think so.
Yeah.
Is it known whether we can't see bioluminescence? Is it known whether when we can't see bioluminescence, it's because it's not happening, or is it our ability to see it?
I would assume the latter, more so it's our ability to see it. It doesn't make sense to me that it just stops, right? Because there's some advantage to it, but of course, maybe if when we can't see it, it's too bright out, and then there's no point of them emitting bioluminescence and wasting energy doing that.
Yeah. Let's see. Are plankton affected by climate change? And if so, how? Or are the plankton?
I mean, yes, I'm not exactly sure in what ways, but I mean, for example, with phytoplankton, at least not considering bioluminescent plankton, we have these huge algae blooms that people always talk about. Like there's certain times of the year, certain places where you're not supposed to swim because there's this massive influx of like nitrogen, and then there's this massive influx of algae. And some of the bioluminescent plankton are part of those red tides and algae blooms. But yeah, so I guess it like is more farm runoff and things like that is in our oceans, then there is like a huge increase.
Well, and I think it's really helpful that you're helping to do a baseline study of bioluminescence in Bellingham Bay, because then we can kind of see how it changes year after year.
Yeah, thank you, Sarah. And I'll just say that for years, I've been collecting plankton samples in the bay to generate the kind of data set that will allow us to assess whether or not climate change is affecting plankton. They're naturally so variable, and an environment like the Salish Sea is so dynamic that to be able to determine exactly what the trajectory of change is with respect to change in the environment requires a lot of data.
Fair. There's a question about being able to follow progress as the study goes. I would suggest joining our newsletter, following us on our social medias. We'll figure out the best way for people to follow along as we collect data throughout the season. We're getting a lot of questions. I'm not sure we're going to be able to get to all of them, just so you guys know, but feel free to email me. I'll write my email in the chat as well. But their next question is, can you get them to luminesce under a microscope? Do you have pictures or video of that?
Robin actually was just sent me an email today of looking at one of the methods of the studies that we looked at. Do you want to talk about that a little more?
Yeah, so you theoretically can do it. We've never done it in my lab, but we're going to try this summer. And it's helpful to use a very fancy microscope called a confocal microscope that uses a laser, and you can kind of do cross sections through a cell. You build a little chamber so that the Noctiluca can, or whatever bioluminescent phytoplankton or whatever bioluminescent plankton fits in the little chamber, and you can change the laser frequency, so you can stimulate the bioluminescence and then switch over and visualize it.
Okay, our next question, is it possible that it could be Noctiluca, but that they're producing bioluminescence through another protein substrate reaction? Or is all bioluminescence definitely only produced with the luciferin reaction?
Well, like in that jellyfish example with the green fluorescent proteins, there definitely is other ways to emit bioluminescence. But I think in the Noctiluca that they looked at with the Gulf of Mexico and then along the West Coast, they were all the same species. So yeah, maybe they could be emitting bioluminescence still with a different strategy, but I assume just because they're the same species that it's unlikely.
Okay, and our next question, have you tried looking at these with other light spectrums like infrared or ultraviolet?
Well, my experiment is only just starting, so I haven't, but that would definitely be interesting to look into.
Maybe those peacock mantis shrimp can see some other.
Yeah, exactly.
And then it looks like our last question for now is, what is the mesh size of the plankton nets you use?
Oh, Robin just answered that.
Oh, perfect.
20 micrometer net.
Awesome. Well, perfect. Well, thank you, everyone. If there's no final questions, one thing I am also going to share is a link to for our donation page. So we want to make sure that Lucy has all the tools and materials for her to be able to complete as many samples as possible this year, as many of those DNA studies, as well as to have some microscopes for our youth programs to, enjoy all of the things that we're seeing. So please consider donating and definitely consider joining us for a bio tour. Once again, I'll share that site as well, but we're excited for a great season.
Thank you so much.
Perfect.
Also, something that Christine, one of our kayak guides, wrote in the chat is that we do private bioluminescent tours too.
We had a bachelorette party come out for one time, and that was kind of fun. Yeah, if anyone wants to hop on with any final questions, feel free to unmute yourself, but thank you so much.
Thank you for your presentation. That was very informative.
Thank you for coming.
Thank you. Thank you.
Thanks, Deb. I'm going to go ahead and end it. Thank you, everybody.
Great job, Lucy. This was wonderful. Thank you so much for hosting, Sarah.
Yes, thank you for organizing it all.
Congrats on a great presentation.
Thank you.
Thank you, everybody. Bye.